Hi, I'm Tracy Fischer. I'm an associate professor with the Tulane National Primate Research Center and the Tulane University School of Medicine. Today I'm going to talk to you about neuropathology of SARS-CoV-2 infected aged non-human primates. Now, as I'm sure that all of you are already aware, SARS-CoV-2 infection is associated largely with respiratory disease. However, early in the pandemic, we recognized that extrapulmonary complications were also a significant concern, including a number of neurological manifestations, which would suggest or indicate that there is some impact on the brain with infection. So in response to the pandemic, very early, even before the CDC or the WHO had actually designated this as a pandemic, we started a pilot study of SARS-CoV-2 infected non-human primates using African green monkeys, which were wild caught from St. Kitts, approximately 16 years old, and included 2 males and 2 females, as well as rhesus macaques, which were from our specific pathogen-free colony here at the Tulane National Primate Research Center. These animals were approximately 13 to 15 years old and included 3 males and 1 female in this pilot study to develop a non-human primate model of infection. Now, the animals were inoculated with what's referred to as the Washington strain. This was the first isolate from the United States, and we used 2 routes of challenge. One was through a multi-route mucosal challenge, which included the conjunctival, nasal, pharyngeal, and intertracheal routes. This was performed on 2 of each species. And we also did an aerosol challenge. Again, 2 of each species were included with this challenge. Now, all animals, regardless of the challenge, established infection within that first week. And this was defined by RT-qPCR of swabs or brushes taken from multiple collection sites. And you can see on the right side of the screen the different areas that we sampled from, including the buccal, nasal, pharyngeal, bronchial, vaginal, and rectal routes. And you can see that all of the animals developed some infection, which was sustained for the majority of the animals throughout the study of approximately 28 days. Now, all of the animals did survive to the study endpoint, except for 2 of our African green animals who had both developed acute respiratory distress syndrome, or ARDS. And this was seen, again, just in the African green monkeys. They're both female, and one had been challenged to the aerosol route and one through the mucosal route, so that did not appear to be the issue for who developed infection, or ARDS. Now, all of the animals had actually demonstrated some degree of pulmonary pathology. The African green monkeys had pathology that ranged from minimal to severe, as you can see with panels A and C. In C, you can see this is from an animal who had developed ARDS, and you can see the significant lesions and pathology within the lung. The rhesus macaques also showed pathology that ranged from minimal to moderate, so they had a relatively less severe disease than what we had seen with the African green monkeys. However, they still did develop disease. All of the animals did have lesions in the lungs that were multifocal and randomly distributed. And this is actually published in the American Journal of Pathology by Robert Blair, who is a veterinary pathologist here at the Tulane National Primate Research Center. Now, all of the animals did have some productive infection in the lung too, as you can see here. The top panels are from the rhesus macaques and the bottom are from the African green monkeys. And you can see this bright pink or deep pink color, which is identifying SARS-CoV-2 nucleocapsid in the lung through immunohistochemistry. Now, of course, our interest was in the brain, and at the time of this investigation, there was very little information coming from humans. So we did a sampling of the brain, looking at the primary lobes, frontal, parietal, occipital, and temporal lobes, the basal ganglia, and cerebellum, as well as the lower brainstem, including the pons and the medulla. Now, our overall hypothesis when we approached this work was that we would see neuroinflammation in the brain. And this is largely based on the fact that we have a significant inflammatory event that occurs in the periphery that we anticipated would translate to the CNS compartment based on earlier work with viral infections. And so our first study included looking at neuroinflammation. We first looked at IBA1, which is a pan microglial marker, and this is seen even in normal brain or non-affected brain, where we would anticipate that the microglia are expressing IBA1. So IBA1 is upregulated in the context of inflammation, and we can see that here in all three panels. In panel A, you can actually see where the morphological changes are occurring in the microglia, as well as some changes in how they are spread through their own territory. So normally microglia like to maintain their own territory, and you'll see them start to cluster up. You can see this in panel A. In panel B, you can see a nodular lesion. And then in C, we saw some accumulation of microglia around blood vessels, suggestive again of inflammation and something happening there at the blood-brain barrier. We also saw upregulation of HLA-DR. This is an MHC class II molecule that is normally in normal brain or non-affected brain we do not detect. However, in the context of infection, we do see upregulation of HLA-DR, and you can see really nicely in panel A those morphological changes that we see in activated microglia where we have retracted and thickened processes. And then you can also see in panel B where they're starting to accumulate in certain areas, and then in panel C you can see this as a nodular lesion. Now, wherever we see microglial activation, we would also anticipate that the astrocytes are also going to be responsive. And so we used a pan-astrocyte marker, a gliofibrillary acidic protein, and performed immunohistochemistry, and you can see both in the rhesus macaques and the African green monkeys. This is areas of the same brain. This is the cerebellum, and you can see in the white matter of the rhesus macaques and the African greens that are infected, we have significant upregulation of GFAP. Astrocytes also, like the microglia, like to retain their own personal domains, and you can see they're losing this. They're starting to accumulate. And then we also see retracted processes in larger cell bodies than we would expect in under physiological conditions. So while this inflammation was very interesting to us in infection, there were some other striking features that kind of shifted our attention into other directions. So one of the most prominent things that we observed when looking at the brain tissue was what appeared to be neuronal injury or even neuronal death. And so what I'm showing you here is, if you look in panel A, or the only panel that we're showing here, this is a row of nice healthy Purkinje cells in the cerebellum. And you can see here in panel A that in the context of infection, we saw large regions of these Purkinje cells with shrunken cell bodies, pycnotic nuclei, where you see highly condensed chromatin that tend to stain or pick up the hematoxylin and stain a very dark blue. It almost appears black. We also saw intracellular vacuolation. You can see here in panel A with the arrows pointing, and then vacuolation of the neuropil. We also saw this in other brain regions. In panel B, we're showing brain stem, where you also see the vacuolation of the neuropil and pycnotic nuclei. You can see the asterisk in there that is indicating a region of that. And also we see shrunken cell bodies in the brain stem. And this is indicative of or suggestive of neuronal death. So we started to explore that a little bit further to confirm or verify that it is in fact death that we're seeing. And so the first thing that we looked at was fluorogade C. This is a special stain that stains degenerating neurons. It's not fully understood how this works, but it is hypothesized to bind to the anionic elements that are within the inner layer of the cell membrane, which would only be exposed in the context of cell death. And you can see these large areas of the Purkinje cells that are staining with the fluorogade C, suggestive of neuronal death or neuronal degeneration. So we also looked at cleaved caspase 3. Now, as you may know, inactive pro-caspase 3 exists in the cytoplasm and is cleaved by initiator caspases such as caspase 8 and 9, regardless of whether it's an intrinsic or extrinsic signal that would indicate a push towards cell death. And so we get this cleavage event and formation of tetramer, which is in its active form. And once we see active caspase 3, this indicates a commitment of the cell to undergo apoptosis. So what we found was that, again, here we have those Purkinje cells in panel A, and you can see the cytoplasm as well as the nucleus showing cleaved caspase 3 positivity by the Purkinje cells, as well as other cells within that Purkinje cell layer. We saw it in other regions as well. On panel B, we're showing a brain stem, and you can see several areas or several cells which appear to be caspase 3 positive, suggesting that these cells are committed to undergoing death, cell death. Now, this was actually a rare feature, myelin vacuolation. It was rarely seen. We only saw it in 2 of the animals. However, this was a relatively small pilot study. So what was interesting about this is that we do have reports of myelin demyelinating disease in the context of infection. So although this was a rare event, we thought it was worth reporting. It's unclear if this is caused by the virus. However, we only did see it in the infected animals in this small study. Now, more frequently, we saw a number of microhemorrhages in the context of infection. You can see this in all of the panels. A through D show infected animals, and E and F are actually our age-matched control animals that underwent the same procedures as our infected animals. And you can see there we have some microhemorrhages in these animals, which does occur with aging. We did quantitate this, and in total brain, we do see a trend towards an increase in the number of microbleeds in the areas of brain that we investigated. This did not reach statistical significance except for in the basal ganglia, where we saw a significant increase of these hemorrhages in the context of infection as compared to our non-infected but age-matched controls. Now, we saw a number of these bleeds that some appeared to be associated maybe with a thrombus, while others did not appear to be. So we wanted to explore this further, and so we performed immunohistochemistry using CD61. This is a factor that participates in platelet aggregation through its complex with CD41 and a number of other factors. And as you can see here, in both the infected and control animals, we had some CD61 positive thrombi in the blood vessel lumen, and you can see that in A through D. However, we also saw some bleeds, which you can see in both E and F, that do not appear to be associated with a thrombus. So we wanted to quantitate this because we saw this in both the infected and the non-infected animals, and our quantitation demonstrated that largely animals that are infected had a greater frequency of microhemorrhages that were not associated with CD61 positivity as compared to our controls. This is true for all of the animals except for AGM5. She was one of our age-matched African green controls, and she had a significant number of bleeds in her brain. As you can see, we have a total of 19 here, and she actually looks a little bit more like our infected animals who had a greater frequency of CD61 negative in the purple versus CD61 positive in the pink bleeds. And this may suggest that we have thrombolytic factors that have removed the thrombus, and so it's not seen in this section, or it could suggest that there are other mechanisms for these bleeds, which we want to explore further. Now, of course, a really important question in all of this is, where is the virus? So in our study, which did not have the olfactory bulb, we did not see virus very frequently in these regions of the brain. We did, in collaboration with Tony Hu at the Tulane School of Medicine, we performed a analysis of virus in whole brain sections, and you can see that these are largely in agreement with rare virus. So it's really just this line above the the bars above this line here, and you can see that the cerebellum, basal ganglia, brainstem, frontal lobe, as well as cerebral spinal fluid only showed virus detection in some animals. And this was largely what we had seen by immunohistochemistry, where sometimes we would see the virus in specific brain regions, while other times we did not in the same animal. So it was really rare. When we did see it, it appears to co-localize with von Willebrand factor, which is an endothelial cell marker, which you can see in the panel to the right, with where you have co-localization of von Willebrand factor and the nucleocapsid of the virus. Now, to gain additional insight into what might be causing this injury, we looked at some of the clinical measures. This was not a study that was set up specifically for the brain, so there still remains a number of questions. However, one thing that we did observe was that all of the animals largely experienced some mild but chronic hypoxemia, as you can see with this reduction in oxygen that was recorded for the majority of the animals. We also saw an increase in CO2, which may suggest impaired gas exchange. We hypothesized that this might lead to tissue hypoxia as well as vascular injury from the hyperimmune response due to the infection, as well as reduced oxygen. And so to begin to explore this question, we looked at hypoxia-inducible factor 1 alpha. So the alpha subunit is normally under normoxic conditions, it would be degraded. However, in the context of hypoxia, this is upregulated, so we get increased transcription of the protein as well as stabilization of the protein where it does not get degraded. And we saw a significant HIF1 alpha upregulation in the context of infection that appeared largely around the vasculature. And we quantitated this. This is through a non-biased quantitation software called HALO, and you can see both in the brainstem and the basal ganglia we had a significant upregulation, whereas in the cerebellum there was a trend towards an increase in HIF1 alpha in the context of infection. However, it did not reach statistical significance. So animal models are only as good as how well they translate to human disease. And among humans, we see similar injury in the brain as what we were seeing in the animals, as well as clinical manifestations that would support what we're finding microscopically in the brain. This includes a number of different types of disease and injury, including cerebral vascular disease, encephalopathy, where patients might, a large number of patients will present with altered mental status or encephalitic presentation, and this was reported with and without confirmatory imaging. A virus in the CNS appears to be rare in the CSF. It's rarely found when it is investigated, at least according to the number of case reports and case series that are out there. Autopsy brain tissue, it has rarely found. However, this is inconclusive, and there may be some areas of brain that are more vulnerable to infection than others. In autopsy brain, we see widespread neuroinflammation with a limited number of nodules and cuffs, is similar to what we had seen in the animals. We see lymphocyte accumulation, but this is generally in the context of encephalitis, which is thankfully rare. And there's a number of reports now that have suggested hypoxic ischemic injury, neuronal injury and loss, as well as microvascular injury in human subjects. So we had done a systematic review and meta-analysis while we were doing our work in the animals to get a better understanding of how the virus might contribute to CNS injury or disease. And so we included 227 different studies in our quantitative analysis, or our meta-analysis in this, and it is reported. This has been published last year in Frontiers of Aging Neuroscience. So, but I will briefly discuss what our findings were. And in this meta-analysis, we actually excluded anybody who may have a pre-existing CNS disease that would confound our understanding of what contribution the virus is actually making to CNS disease. So of these reports, it included 2,390 subjects, and you can see we categorize their different manifestations and these different categories: cerebral vascular disease, peripheral neuropathies, encephalopathies, demyelinating disease, smell and or taste disorders, CNS inflammatory disease, and then we made a category of other where symptoms were described. However, they did not necessarily fall under these other categories of disease. And what we found was that while smell and or taste disorders were the most frequently observed or reported, cerebrovascular disease was second, and that was a pretty significant population in our analysis. When we stratified this by age, we see in individuals over at 50 years old, this is a significant risk for cerebral vascular disease in the context of infection, whereas smell and or taste disorders appear to be more prominent among younger individuals. However, it's important to note that some severe, critical, urgent CNS manifestations such as cerebral vascular disease are also reported in young individuals, including those under 19 years. So we correlated these different manifestations with age and found that cerebral vascular disease had a positive correlation with increased age. However, CNS inflammatory disease, taste and or smell disorders, and our category of other were the only other categories associated significantly with age. However, this was an inverse correlation. Now, these are correlation matrices that show the relationship of age with cerebral vascular disease, or the different manifestations. Cerebral vascular disease is the only one that, again, was associated with an increase in age. Panel B shows that we have a higher risk for severe or critical disease with age. And then panel C just shows that we have a increase for comorbidities that are associated with a severe or critical disease outcome. And these are higher among individuals who are aged. We do not really see any sex differences, which are included in panels D through F. Now this puts it all together where we have age and sex and how it's associating with these different neurological disease categories, and you can see the disease severity or the comorbidities did not necessarily correlate significantly with any particular neurological disease outcome, although some were more put an individual at a greater risk for certain types of manifestations. So in conclusion, we showed that infected African green monkeys and rhesus macaques are highly relevant animal models of SARS-CoV-2 infection. They can recapture what human disease and autopsy findings are showing, and they also reveal brain injury even in animals that did not develop severe respiratory disease. And this is a really important outcome of this study, where we were able to actually look at the brain microscopically in animals that would not have succumbed to disease, whereas in humans we are only able to look at the brain in this manner after death. So this is important, as this really shows neuropathology may be a common complication of infection. We also saw varying degree of neuropathology, including neuroinflammation, neurodegeneration, microbleeds, a number of microhemorrhages, and, as mentioned, infrequent myelin vacuolation. Now, I just want to leave you with how this might be important for individuals who experience neurological symptoms of post-acute sequelae of COVID-19, or PASC, which is also referred to as long COVID. Whereas this is going to, the neurological manifestations likely reflect what area of the brain is impacted, the extent of the injury, and whether we get a complete resolution of injury. And this is as an example, I am pointing to a study from 2021 of imaging individuals who had experienced persistent impaired smell. And these individuals showed a reduction in the metabolic activity in the bilateral limbic cortices, and I'm showing that in the top picture, what that area of the brain would include, and the left insular cortex. And again, I'm showing that here in the lower image. Now, there's also the question of whether there's virus in the brain, and if so, what cells are infected, and do we get a resolution of this infection in the brain, or is it an abortive infection that could still impact the function of the cells that it's infecting? And then whether or not we have a prolonged and or augmented neuroinflammation that does not get resolved, and how this might contribute to pathological brain aging. So in conclusion, I'd like to acknowledge all of the individuals who participated in this work. This was a huge undertaking by a wonderful team of veterinarians and scientists, and especially want to recognize the efforts put out by the individuals in my lab, Lynn Helmers and Meredith Mayer, that did the bulk of this work, which is really labor intensive. So I'd also like to acknowledge our funding from the NIH, the FASC grants for COVID-19, and Tulane University. Thank you very much.